Good morning and welcome to today's webinar presented by RedShift BioSciences and Pressure BioSciences. My name is Matthew McGann and I'm the Field Applications and Marketing Manager at RedShift Bio. Today's webinar is going to be focused on the next generation IR spectroscopy of protein structure perturbed by pressure. And this morning I'm going to introduce to you microfluidic modulation spectroscopy as a tool for measuring higher order structure of proteins in conjunction with pressure perturbation. Measuring protein structure can be a complex task that requires an understanding of many different analytical techniques and a broad understanding of how protein structure, function, and formulation can affect one another. As we know, protein structure can be very sensitive to changes in local environments, such as pH, ionic strength, temperature, and even sample concentration. When working in applications such as formulation and development for biopharmaceuticals, a full understanding of protein structure is really a two-sided coin. On one side, we have the need to fully quantify and measure the structure of a protein itself. And on the other side, we have the need to ensure that structure does not change during manufacturing, processing, fill finish, shipping, and delivery of the sample all the way to the patient. In this talk, I would like to introduce you to the AQS3 Pro, an instrument which is a new generation microfluidic based IR spectrometer with functionality far beyond that of existing technologies. To begin with, I'd like to discuss some of the current limitations of the toolset used for protein assessment. When it comes to biotherapeutic products, there are four primary areas of characterization that is generally required by a regulatory body, and these four areas would come together to form the fingerprint of a biological molecule. This fingerprint would include quantification or a measure of the concentration, structural information, including secondary and tertiary structural information, stability profile information, including the effect of excipients and additives to the sample, and an aggregation profile covering particles from oligomers to submicron and subvisible particle content. When it comes to quantification, UV-Vis spectrophotometry would be considered the most incumbent technique. We do know that UV-Vis spectrophotometers have quite a limited dynamic range, and it is often necessary to dilute samples in order to measure the concentration accurately. In terms of structural information, measuring secondary structure, circular dichroism would be considered the most incumbent technique. However, circular dichroism does have quite significant limitations, both in terms of the concentration range that it can measure in, which is quite low, and its ability to measure in complex formulations, as it is not really compatible with excipients. In terms of stability, differential scanning calorimetry would be considered the gold standard for understanding the thermodynamic properties of a protein as it undergoes unfolding processes. DSC, however, does not provide higher order structural information. With regard to aggregation, size exclusion chromatography and analytical ultracentrifugation are both methods that can be quite complicated to implement. However, some of them do provide automation in order to complete the measurements. So if we break down protein characterization into the four areas of protein structure, we can see that we have a wide and diverse array of tools available to us for characterizing proteins. In relation to protein primary structure, mass spectrometry offers a high throughput technique that allows us to characterize the mass of proteins very rapidly. In terms of tertiary structure, size exclusion chromatography allows us to calculate the molecular weight of our proteins in a very high throughput fashion. Quaternary structure can easily be calculated using techniques like dynamic light scattering, and these techniques are also very high throughput. The issue, however, is in relation to secondary structure. When it comes to measuring secondary structure, we don't have the same level of automation and breadth of tools available. Circular dichroism would be the most incumbent technique used for measuring protein secondary structure. And we know that the concentration range of circular dichroism is quite limited, and its ability to measure samples in complex buffers or with excipients is very limited as well. FTIR, on the other hand, has a better ability to deal with excipients and complex buffer systems, but requires a much higher concentration in order to obtain a good clean signal, and is quite difficult in terms of reproducibility and operator usability. Microfluidic modulation spectroscopy is a new technology which can assess protein secondary structure over a concentration range from about 0.1 mg per ml to over 200 mg per ml, and is compatible with the complex formulations required to manufacture stable biopharmaceutical products. In comparison to the incumbent technologies such as CD, DSC, and FTIR, the system can cover a dynamic range of 4 decades of concentration, which is far beyond the capabilities of these techniques. The AQS3 Pro system is the next generation of instruments to assess protein secondary structure. The system combines 4 key features. Firstly, a high-powered quantum cascade laser, providing significantly brighter light source than a traditional spectrometer. A high-precision microfluidic flow cell, providing a unique microfluidic modulation spectroscopy platform that minimizes background drift and other issues associated with benchtop spectrometers. A fully automated sample handling system, which is compatible with standard microwell plates for easy unattended measurement. And finally, a user-friendly software platform that provides simple setup of measurements and advanced analytical tools that allow you to rapidly determine higher order structure information for easy sample comparison. At the heart of the AQS3 Pro system is the microfluidic flow cell. The microfluidic flow cell, once primed with sample, can rapidly alternate the buffer and sample through the observation zone, providing an easy measure of the differential absorbance between the sample and buffer. Interpolation of these data points can provide a spectrum. The rapid sample buffer referencing technique without moving the cell provides spectral reproducibility of greater than 99%. One of the major features of the AQS3 Pro is its analytics software package that allows you to convert spectra all the way through to higher order structural information with just a few clicks of the mouse button. To highlight this, we're going to go through an example workflow where we've taken an antibody fragment and exposed it to a range of temperatures in order to look at essentially a melting profile of that sample. Before we do that, if we have a look at the lower set of graphs on this screen, you can see that because this is an absorbance-based measurement, it is possible to generate a concentration curve of our samples. And one of the great features of this system is that the measurable concentration range, as I've mentioned, is between 0.1 and 200 mg per ml, and the system shows great linearity across that range. Moving on to the upper workflow on this screen, it's very easy to take this differential absorbance spectra and convert it into higher order structural information, as we'll discuss on the next slide. Now looking at this slide, if we follow the top line of spectral processing, what you can see here is that we've taken the differential absorbance spectrum and converted it to a second derivative in this case. The second derivative is a great way of easily identifying differences in spectra that may not be as obvious in the raw spectrum. Once we have a second derivative, we can then produce what we call a difference spectra. Now this is subtracting each spectrum from a reference spectrum, and it allows you to very quickly highlight the key wave numbers in which significant change is occurring in the sample. Once you know these key wave numbers and can relate them back to structural parameters, it is then possible to generate a stability profile. In this case, we've tracked 3 wave numbers related to beta sheet composition of the sample, Those wave numbers are 1624, 1641, and 1697. And we've also traced an alpha helix parameter at wave number 1654. And what this graph clearly shows is that the structural composition of this sample changes drastically once the sample reaches approximately 65 to 70 degrees. Following the lower path in this software workflow, we can also generate higher order structural information in terms of the fractional content of higher order structure very quickly and easily by taking the second derivative and converting it into an area of overlap plot, which involves inversing the plot and then applying a global baseline. We can very quickly look at the percentage area of overlap between two spectra to determine how similar or how different those spectra are to one another on a simple percentage basis. Once we have these spectra, we can then apply some simple models in terms of higher order structure to deconvolute these spectra, which allows us to calculate the fractional contribution of a range of structural parameters such as beta sheet, antiparallel beta sheet, alpha helix, and so forth. In the final plot, what you can see here is that we are graphing the composition of each of these higher order structural features relative to the temperature that the sample has been exposed to. Now, knowing that red represents the highest temperature, you can see that as the sample is exposed to higher temperature in this case, the amount of antiparallel beta sheet increases, and conversely, the amount of beta sheet content decreases. Now, this is very typical and expected of an antibody type sample. So to conclude this section of this morning's webinar, I'd like to review some of the key pieces of information relating to the AQS3 Pro. The system provides 5 critical protein quality measurements, those being aggregation, quantitation, stability, similarity, and structure. With the improvements in light source, the system provides a 30 times improvement in sensitivity relative to current conventional infrared systems. It also offers the widest measurable concentration range of any structural analysis platform, measuring from 0.1 mg per mil to greater than 200 mg per mil. With the integrated autosampler, the system provides multi-sample capability and can lead to a 20 times savings in direct labor costs due to the ability to operate the instrument hands-free. Finally, the automated protein analytics tools provided by our software platform directly fulfill the needs of both the operator for simple operation and the scientist for simple data interpretation to take you from your sample to your fully processed data in as short a period of time as possible. I'd now like to hand the webinar over to Alex from Pressure BioSciences, who's going to introduce you to his system and then show some examples of how the AQS3 Pro can be used to observe protein structure under pressure.
Hello everyone. I'd like now to switch gears and talk about one of my favorite subjects, thermodynamics. But please don't worry, I have no equations in any of my slides. First, I'd like to start with a couple of citations from a classic 1999 publication by the champions of high-pressure protein refolding, 2 University of Colorado professors, John Carpenter and Theodore Randolph. They said, protein aggregation is subject of intense investigation in disciplines including human medicine, fundamental protein chemistry, and biotechnology. For example, aggregation could have severe consequences in human disease, such as Alzheimer's disease and Parkinson's disease, and in the manufacturing, shipping, storage, and delivery of protein therapeutics. They also said that in spite of recent progress in theoretical and computational approaches to understand protein folding and refolding, efforts to manipulate folding in vitro often are plagued by competing off-pathway aggregation processes. The bottom line of it all is that protein aggregates are highly immunogenic, potentially causing adverse immune reactions and inactivation of biologic therapeutic agents. Understanding and control of protein aggregation can significantly improve our ability to produce better biologics. At Pressure BioSciences, we employ high hydrostatic pressure to control protein conformation. Most pressure effects in biological systems are facilitated by hydration. Proteins undergo unfolding by infusion of water molecules into their hydrophobic core. Such unfolding is typically reversible at moderate levels of pressure. I'd like to stress specifically that the extent of unfolding by pressure is protein-specific and highly reproducible, much unlike entropic thermal denaturation. Pressure allows us to manipulate protein structure with no or minimal addition of chemical or thermal energy. Pressure causes dissociation of multimeric protein complexes. As we apply higher pressure, it leads to unfolding of monomeric proteins, resulting in modulations of protein function. Pressure happens to work in synergy with chemical denaturants. In the next few slides, I'm going to show you a few examples of how pressure can be used in a convenient research settings to study proteins. At the beginning of the 20th century, Harvard University professor Percy Bridgman laid the foundation for today's high-pressure solid-state physics. He also experimented with so-called soft matter, which is the way physicists refer to biological material. He noticed that an egg white could be cooked at room temperature solely by application of high pressure. We have recently repeated this classic experiment in one of our instruments. Pressure levels generally higher than 500 megapascal or 70,000 psi begin to cause irreversible protein denaturation. Longer exposure or higher levels of pressure lead to more rapid and more complete denaturation. Pressure denaturation of proteins is synergistic but very different mechanistically from the thermal denaturation. Thermal energy promotes random movement of protein molecules until the alignment breaks, while pressure actually suppresses Brownian motion and drives every protein molecule to the same conformational state by infusion of water molecules into what normally are solvent inaccessible cavities inside protein structure. Pressure effects were known for more than a century. However, all this time, high-pressure research was confined in the laboratories of a few enthusiasts because there was no commercial equipment available to generate and control very high level of pressure in the life science research settings. Our company has introduced a line of dedicated appliance-like benchtop high-pressure instruments that help to promote high pressure as a convenient research tool. Pictured here is the Barocycler 2320 Extreme, an appliance that controls both temperature and pressure and allows up to 16 samples of up to 150 microliters each to be pressurized in one batch in specialized sample containers. Sophisticated software allows accurate pressure control ranging from continuous pressurization to rapid pulses of pressure from atmospheric to very high level and back to facilitate various biochemical processes, from rapid proteolytic digestion to screening for best chemical environment for protein refolding. We have originally started in the area of biomarker discovery and proteomics by accelerating and improving reproducibility of proteolytic digestion, as I mentioned before. By unfolding protein substrates, pressure helps to expose hidden cleavage sites to the enzyme. Proteases appear to be more resistant to high-pressure denaturation, and several of them, such as chymotrypsin and most notably endoproteinase Lys-C, show remarkable ability to work under pressure like all of us. They actually get significantly accelerated without losing their specificity. In this example, myoglobin as a model system was digested and monitored by disappearance of the parent band on an SDS page gel. Indeed, chemotropic agents and pressure are synergistic. Apparently, pressure makes ordinary organic solvents act as chemotrops too. This is a great news for mass spectrometry community that is generally trying to stay away from any chemical additives in their samples. However, not every enzyme accelerates under pressure. In fact, partial unfolding is more likely to make enzymes less efficient. A yeast lysate was interrogated with the specific chromogenic and fluorogenic substrates to demonstrate the ability of pressure to selectively inactivate various enzymes in their optimized buffers. As you can see here, Alanine S-transferase is shown to be the most pressure-labile enzyme, while the alkaline phosphatase is the most robust enzyme with respect to pressure inactivation. Pressure helps to make fast and reproducible protein digest for peptide fingerprinting in biopharmaceutical quality control settings. It also turns out that pressure helps to digest native unreduced monoclonal antibodies and other proteins in order to rapidly and reproducibly characterize location of disulfide bonds. Pressure also helps to accelerate deglycosylation using protein N-glycosidase, abbreviated as PNGF enzyme. In addition to saving significant amount of time, this sample preparation methods carried out in the Barocycler are shown to be extremely reproducible. This slide shows the work published by a Genentech team that reports pressure-enabled fast Lys-C digestion of their monoclonal antibody product, preserving disulfide bonds, minimizing disulfide scrambling, and maximizing quantitative peptide recovery, thus streamlining multiple critical quality attributes analysis. Now let's get back to protein refolding for a moment. About a third of the total volume of protein therapeutics is being produced in bacterial expression systems. Bacteria do not require expensive media components. They grow very fast in traditional stainless steel bioprocess equipment and produce large amounts of recombinant proteins. With rising demand for biosimilars, antibody drug conjugates, and some fusion proteins that cannot even be produced in the eukaryotes, bacterial expression systems are gaining renewed interest again. Research reagent proteins are also produced predominantly in bacterial systems. Usually overexpressed proteins get dumped into a misfolded aggregated state called inclusion bodies. While inclusion bodies contain mainly protein of interest and can be obtained by a few centrifugation steps, thus making purification very easy, the protein must be solubilized and refolded at the end. Turns out that aggregation mechanisms responsible for protein deposition in inclusion bodies and also amyloid fibrils have a lot in common. Studies done by FTIR demonstrate that both types of protein aggregates are rich in anti-parallel beta sheet structures. Both types of aggregates can be selectively disaggregated and solubilized by hydrostatic pressure. Proteins deposited in inclusion bodies must first be unfolded, untangled, and solubilized to its monomeric state. Afterwards, individual molecules are allowed to refold, guided by appropriate chemical environment in the refolding buffer. Very few proteins refold spontaneously after they are solubilized from inclusion bodies. So typically, protein refolding methods rely on very high concentrations of chemical denaturants such as urea or guanidine that must be removed in a lengthy dilution or dialysis steps prior to refolding. At this stage, reaggregation is likely to occur since unfolded proteins are the most susceptible to aggregation. However, if pressure is used to unfold proteins, it could be simply and quickly turned off when appropriate. Therefore, high-pressure refolding could be generally carried out much faster and at a significantly higher concentrations, leading to greater yields of properly folded active proteins of interest. This slide shows the popular energy landscape of protein folding and aggregation diagram. It shows minimal energy states that sometimes correspond to non-native structures such as amorphous aggregates or amyloid fibrils that could perhaps be even more energetically favorable than native protein structures. However, if one applies the pressure to some of these structures, refolding could be carried out. Previous studies have shown that the moderate high pressure of 100 to 300 megapascal is generally effective for the dissociation of protein oligomers and aggregates, while relatively higher pressure is needed to denature native monomeric protein structures. Pressure propagates through media with the speed of sound and affects every molecule in solution to the same extent. Thus, uniform refolding process could be achieved if chemistry surrounding the protein molecules is right. For example, complete solubilization of human proinsulin inclusion bodies occurs in only 5 minutes under pressure, as you can see the photograph on the left. After the pressure is lowered back to atmospheric level in a 2-hour gradual ramp, and in the optimal redox buffer containing 9:1 ratio of cysteine to cysteine, proinsulin monomers refolds to its active state, and insulin precursor protein, as shown on the reverse phase HPLC chromatogram on the right side. Pressure unfolds inclusion bodies very easily. However, protein refolding requires a lot of other parameters to be just right. Optimization of the refolding chemistry requires a series of multivariant screening and experiments designed to determine correct pH, ionic strength, temperature, concentration of other reagents, co-solvents, and affinity binding ligands, if applicable. Pressure BioSciences offer contract service and equipment for optimization of refolding parameters at the customer site. Our flagship product, Barocycler 2320EXT, has a ramp mode option in its software. This feature allows users to slowly ramp the pressure up, holding it for minutes to hours, if necessary, and slowly release pressure at a predefined rate. This feature is exceptionally useful for protein refolding to prevent rapid reaggregation of partially unfolded proteins. By analogy, protein aggregation is another major problem in the biopharmaceutical industry. Protein aggregates tend to be immunogenic, leading to drug inactivation and unwanted adverse reactions in patients. Pressure could be used as a convenient tool for protein disaggregation. From biomanufacturing to formulations, accelerated stability studies can be carried out to determine whether the protein is happy in a specific chemical environment, or it is about to form fibrils or highly immunogenic aggregates. Understanding mechanism of protein aggregation can help to develop formulations that favor protein stability. Unfortunately, there is no universal method to study protein structure in a rapid manner. Here is an incomplete list of complementary tools that help us to tease out structure of the proteins. While NMR, crystallography, and mass spectrometry provide information-rich content, they cannot be used as a screening tool. Therefore, we're always on the lookout for rapid and automated tools to characterize conformational changes in proteins. Moreover, partially unfolded proteins tend to be very sticky. They aggregate, clog HPLC columns, create turbidity that interferes with fluorescence, UV-Vis, and CD spectroscopy, as well as introduce significant errors in laser scattering measurements. Therefore, we turn to the infrared spectroscopy as a tool that is less sensitive to turbidity and scattering, yet as informative as circular dichroism spectroscopy with respect to protein higher order structure. Thanks to our collaborators at RedShift Bio, we recently got to try their AQS3 Pro system, employing a new approach to infrared spectroscopy entitled Microfluidic Modulation Spectroscopy. As Matt showed a few minutes ago, this instrument can qualitatively report changes in protein structure as a function of time or chemical environment with great reproducibility and high sensitivity. So we thought, what about pressure? For our first set of experiments, in order to understand the power of their approach, we selected a well-characterized model, Human IgG, a well-soluble protein that is known for its propensity to aggregate in specific conditions, pH, temperature, and pressure, all known to trigger IgG aggregation. We have compared effects of 4 levels of pressure and 1 heat condition on IgG aggregation in 2 buffers, conventional phosphate buffer saline and PBS containing 10% N-propanol as a co-solvent. The graph on the right shows Bradford protein assay of protein remaining in solution after pressure treatment. It turns out that our favorite N-propanol acts in synergy with pressure, causing efficient unfolding in irreversible protein aggregation at much lower pressure levels compared to PBS alone. After being pressurized to 60,000 psi, we have lost almost half of the protein in the buffer containing N-propanol, while IgG content was totally unaffected in a similar pressure in PBS alone. We had to discard remaining samples treated at 80 degrees Celsius or 90,000 psi that lost most of the protein to a pellet. Remaining samples were analyzed in the RedShift biosystem. Although all proteins remain in solution, the 60,000 psi treatment of IgG in PBS resulted in a significant, about 6.5 to 7% conversion of the parallel beta sheet content into the anti-parallel beta sheet. This conversion is a good indication of initial stages of aggregation. However, thermal treatment at 80 degrees Celsius resulted in no noticeable structural change. Observed changes remained very consistent for 2 days after the treatment. Even much more remarkable changes were observed in samples containing co-solvent. While samples of 3 replicates for every condition were essentially superimposable, each increase in pressure showed pronounced and proportional conversion of the parallel beta sheet into the antiparallel. Moreover, formation of extra alpha helical regions from turns were also observed, consistent with the literature data suggesting that the aliphatic alcohol co-solvents are conducive to formation of extra alpha helical structures. This is another representation of the same data set on the AQS3 platform. Again, a gradual drop in parallel beta sheet and proportional increase in the anti-parallel beta sheet, while a gradual decrease in turn signal corresponding to increase in alpha helical structures is shown here. Please note that the 60,000 psi sample has been virtually visibly turbid, yet data was completely unaffected by this fact. I hope that I was able to give you a little hint today that high pressure in combination with the microfluidic modulation spectroscopy platform could serve as a powerful platform for protein refolding optimization and studies of protein stability in formulations development. I'm convinced that AQS3 platform could be a very useful tool in our team's toolbox as we work with partially unfolded and aggregating proteins. In the end, I'd like to thank everyone who worked on this project and made today's presentation possible, as well as Genetic Engineering News team for hosting us today. And most importantly, thank you very much, all of you listeners, for your attention today.